My name is Candace Price, and I'm the Bioanalytical Sample Prep Product Manager for MilliporeSigma, and that's the life science business of Merck KGaA. So I think the most innovative technology I'm working on right now would be BioSPME, or Biocompatible solid phase microextraction. And what these are there's single devices that do both extraction and sample preparation simultaneously. And so they come in a few different devices. So one of them would be an in vivo needle probe, and those are meant for sampling from live animals such as mice and rats. And then there's also an in vitro tip device, which is more meant for aliquoted samples, say in a 96-well collection plate or stored in vials or something like that. And the commonality between all of these devices is they are all meant for sample extraction and cleanup for primarily biological samples for either downstream LC-MS or direct MS desorption. And what they're made out of is actually a metal core, and it's coated with a coated silica. And on the coated silica, there's selective phase chemistries that can target either a very narrow range of analytes of interest or a very broad range. So it's complementary to the analytes of interest that you're looking at. And so the way it works is via direct immersion. So you're either going to directly immerse the fiber right into your live animal subject or right into your aliquoted sample. And while the fiber is immersed, the larger macromolecules are actually being repelled by the patented binder that we have on the fiber, whereas your small analytes of interest are going to bind onto the fiber. So once you pull the fiber out of the sample, you just do a quick rinse, and you have a entirely cleaned up sample that's ready for downstream LC-MS or direct MS analysis. And one other thing is it's not an exhaustive technique either. The fiber is really small, so the amount of sample that it's actually extracting onto it, it makes it so that you can do serial sampling or multiple sampling from the same aliquot or from the same subject, because it's not changing the system or the aliquot at all. So one of the main advantages, when I'm talking about how it's repelling the larger macromolecules but retaining the small analytes of interest, so that lends itself really well to free analyte fraction analysis, since all of the protein-bound analytes are not going to bind onto the fiber. Another advantage is the ability to do multi-residue analysis. We actually have a poster at EBF here where I'm presenting on BioSPME for multi-residue analysis compared to traditional SPE and protein precipitation. And so we show that there's better matrix interference removal using BioSPME compared with SPE or protein precipitation, which are two very common techniques that customers use to do sample prep. The biggest advantages are really going to be in time and cost savings with a much more simplified workflow. It kind of depends on the device. So when you're talking about the in vivo needle probes, it's more to do with ethical considerations and the dramatic reduction that you can see in lab animals, again, because you could do serial sampling from the same animal, so that would theoretically dramatically reduce the number of lab animals that you would need per experiment. For the in vitro devices, again, it's really time and high throughput advantages, as well as we do a lot of comparisons with different technologies. And so one of these is rapid equilibrium dialysis, and that would be used for protein binding studies. And so rapid equilibrium dialysis is a commonly used technique today, but the workflow it can be over 8 hours long. And so we show in a lot of the studies that we've done that we can actually use BioSPME for this instead, and we can crunch that workflow down to 1 hour instead of 8. So it's just a huge time savings. And then also the data as well, the precision and accuracy of the data. Pretty much every technology that we've compared BioSPME to, either the data is equivalent or comparable, or it's better in terms of matrix removal and precision and accuracy. So I think there are really infinite possibilities for the technique in general. But I think some of the bigger areas are going to be for the in vivo in the area of PK or pharmacokinetic studies. Again, being able to do serial sampling from the same animal, you could theoretically just use one mouse for an entire time point curve for a PK study rather than using 12 or 18 mice. And then for the in vitro, since that's more of a high throughput area, maybe more for automation, I think that would be a bigger player in more diagnostic assays for clinical. And then also, again, for the protein binding studies that are commonly done in pharmaceutical drug development. And then for direct MS, again, since both of these device platforms can be used for direct absorption into the mass spec, a lot of high throughput screening that doesn't require chromatographic separation. So for things that you want to screen really quick just for a yes or no answer, I think that would be another big area as well. We try to really focus on communication and collaboration with partners and customers. So especially with really disruptive and innovative technologies, they can be really difficult for customers to implement just because of all of the changes that needed to be made and regulatory considerations. And so we really try to maintain really strong technical service and technical support so that we can really help, guide the customer through implementation. We help with method development and just really trying to take a really hands-on approach so that we can make sure the customer gets up and running as smoothly as possible.